Hello everyone, my name is Fatih. I'm a fourth year PhD student in statistics and I am working with Stuart Barber and Robert Aykroyd. Today I'm going to be giving a talk about Voronoi tessellation-based lifting scheme in two-dimensional bounded regions. Since I'm not in Leeds today, I recorded my video, and if you have questions, you can email me, which I will give my email at the end of the slides. So moving on the slides, maybe I can turn off my camera now. So I would like to start my talk with a discussion about the Voronoi tessellation. I would like to give a brief explanation of the Voronoi tessellation. So the Voronoi tessellation is a method to divide the space into smaller regions. So this space can be in 1, 2, or 3-dimensional spaces, but we are interested in 2-dimensional spaces in this study. And there are a lot of application areas of Voronoi tessellations, such as astronomy, ecology, meteorology, and so on. And it's also used in the analysis of spatial data, such as spline methods or intensity estimation. So in my thesis, I was interested in working on Voronoi tessellation in two-dimensional bounded regions. I investigated the statistical properties of the Voronoi cells, and these properties are the areas, the perimeter, and the number of edges of the Voronoi cells, which I'm going to show you in the next slide. And we investigate the boundary effects on the Voronoi cells, and we are proposing a method that can reduce the boundary effects. So the Voronoi tessellation looks like this. We have a set of points in two-dimensional space, and the Voronoi tessellation divides the space into smaller subregions. So these polygons are called Voronoi cells, and the cell edges are perpendicular bisectors between 2 points. So we don't see any boundary on this example. You can think of these points are on a larger space, but we just sampled some of them in the interior space. But we are interested in what happens when boundaries are imposed on the Voronoi cells. So the plot on the left side is the previous plot. And this is the same Voronoi tessellation, but we impose a boundary that is this vertical line, and we observe the changes on the Voronoi cells on the right side. So we see that the shapes of the cells changed, especially for the ones that are closer to the boundary. So the blue points intersect the boundary. When I say intersect, that means that that cell has a vertex on the boundary line. But more interestingly, we have some red points. These points didn't intersect the boundary before it's imposed, and it still doesn't intersect the boundary. But we see that their shapes are also changed. So it's a bit natural to have a change in the areas on the sizes of these cells, which are shown with the blue points. But the red points doesn't intersect the boundary, but still affected by the boundary. So in my study, I investigated how the boundaries are affecting the properties of these cells, such as the cell area, cell perimeter, or the number of these cell edges. I performed a simulation to investigate this. So we generate homogeneous Poisson points. That means we are generating a set of points where the number of points is following a Poisson distribution with a positive finite intensity. And then we restrict that region with a boundary, which we use unit square. And then we observe the cell area, cell perimeter, and number of cell edges in this region. So we repeat the generation of new realizations of points for 1 million times, and from each realization we randomly sample a point, and then we gather all of the information that we obtain from these cells and study how these properties are different. So for 1 million points that we studied, we use their relative locations, and we created these plots. The left plots are showing the cell area near the corners and the edges of the boundary. So this is just a quadrant of the unit square. So we observe the symmetric properties on each side of the each part of the unit square. So this is from 0 to 0.5, but we actually this continues on the right hand side, and for the top part as well. So we see that the cells very close to the corner are having the smallest area, and the ones that are closer to the near edges that are having still smaller area, but it is smallest at the corner. And then we summarized these properties based on the surface transects on the right-hand side plots. We took a diagonal transect. That means that we followed this direction and checked how the cell properties change on this direction. And the middle says a central transect, for example, starting from 0.3, and then we take a horizontal transect, or it can also be a vertical transect in different places, for example, 0.4 as well. And we use the edge transect, that is a transect that is very close to the edge. And we see that the cell properties change proximity to the boundary. And this is another boundary type, which we use the convex hole as the boundary. So we have even smaller areas near the corners of the region. So we also approximated the cell properties for different boundary cases. So if there is no boundary, the cell properties are approximated using the 3-parameter gamma distribution, and these are the parameter estimates. And we have 3 cases: the absence of the boundary, where we consider these Voronoi cells as if it is in an infinite plane, or when we use unit square boundary and convex hole boundary, we see that the parameter estimates are different. So the cell properties are different. So we have area, perimeter, and edges, and summarized all of these properties. So the conclusions from this study is the distribution of the cell properties are different in the absence and presence of the boundaries. And these differences are changing proximity to the boundary, and it is valid for different point intensities as well, which we check the very different intensities, lower than 200 and higher than 200. And the key conclusion from this study is the observed cell properties, when there are boundaries, do not reflect the true cell properties. And this work is published in our 2021 paper. If you are interested in the details, you may have a look at it. So on the other hand, I also work on the lifting scheme. That is a separate topic than the Voronoi tessellation, but the Voronoi tessellation is used inside the lifting scheme. So we call it as the Voronoi tessellation-based lifting method. The lifting method itself is a method to denoise the noisy data based on the irregularly spaced points. So it works as a denoising method for irregularly spaced data. And when I say denoising, that means that we have a data, but we don't have a clear pattern of the data. So we separate the noise and estimate the underlying true patterns in the data. And this lifting scheme is a second generation wavelet method. If you are familiar with the wavelet methods, this method is related, and the background of lifting is built upon the wavelets, but it relaxes some of the features of the wavelet methods. So it's called as the second generation wavelet methods. And we use the Voronoi tessellation in the lifting method to determine the neighborhood structure of the points and the lifting order and the prediction of some weights when we are having the estimates. And the lifting method assumes a model, yi, that is the observed data, but the observed data has an underlying pattern that is a true fxi values and plus the noise, which is a Gaussian noise. And we observe yi at the data locations, and we would like to estimate the underlying pattern f hat I. And we use the method introduced by Jensen. It's called lifting one coefficient at a time. That means that given the data, noisy data yi, we transform this data to a set of coefficients, and then we threshold these coefficients. The thresholding method works as we identify the coefficients that are due to the noise and that are due to the true patterns in the data. So it is a transform that creates the coefficients d that is actually calculated by using a transform matrix, which I'm not going to go into details, but you can ask me if you have any questions about it. So the true, the observed data, the noisy data, it's a vector of y, is multiplied with the transform matrix, and we obtain the coefficients d. It's a vector of coefficients. That coefficients represent very small coefficients and large coefficients, and we assume that the small coefficients are due to the noise, and the large coefficients are due to the true patterns in the data. So what we do is we threshold these coefficients and set the small ones to 0. And then we apply the inverse coefficients that actually gives the estimate of the underlying true pattern f hat I. If we don't do any the thresholding on the coefficients, it's directly gonna output the noisy data values y actually. So the function estimation is made using this transform matrix and inverse transform of the thresholded coefficients. So I'm not going to move on the details too much on this lifting topic due to the time, but I would like to give a few ideas on this. So when we are estimating the underlying true patterns, we use some weight methods that are calculated from the Voronoi tessellation cell area. But these cell areas in the bounded regions do not reflect the cell area, true cell area. So we propose a method that we can predict the true cell area when the data is given in a bounded region. So we use regression-based methods, and we, given the other properties of the cells, which are the predictors, we predict the true cell area as if there is no boundary. So we use the cell area obtained from the convex hull boundary and unit square boundary and the prediction of the boundary. These are 3 different cases. So we use all of the different types of area from these boundary conditions and the predicted area, and perform the lifting and see how the lifting works. Lifting estimations work in the function estimation. So we rely on different types of point patterns, which are, some of them are regular point pattern and homogeneous and clustered point patterns, and the ones between are different degrees of regularity and clustering. It is probably more clear to express what I meant in the lifting scheme and the weight methods. So we perform the lifting scheme to estimate an underlying true pattern in the function. So this plot is a function called Heaviside, a two-dimensional test function. So what we do is we are given this true function. We have this true the formula of this true function, and we add the noise separately. So we add the noise, but we already know the true values. And then we apply the lifting method to separate the noise from the data. When we are applying the lifting method, we are using different weights that are calculated, that are obtained from the Voronoi cell area. So we use different weight methods: cell area obtained from convex hull, cell area obtained from a unit square boundary. And these are some types of adjusted weight methods. So these are different types of predicted cell area for different models, using different models. I'm not going to go too much into detail, but we can just categorize the red ones as the ones that are directly obtained from the cell area bound due to the boundary, and the last four are different types of predicted cell area. So when we perform the lifting scheme using these alternative weight methods, we see that we separated the results for interior region and the edge region. So the interior region will be a smaller square in this unit square where the points are not really affected by the boundaries. But the edge region is the outer region, so where we are more interested in. So in the edge region, when we check the function estimates, we see that our proposed area prediction method is giving the smallest mean squared error on the function estimation. And usually these two different or this open circle and triangles are very competitive results, but for the more clustered points, actually these modified versions of these models, the star star methods are the modified versions of these blue circle and the triangle. In the clustered point cases, the solid circle and triangle are giving better estimates. So we tried this for different different test functions as well. So we see for this bumps function, we again add the noise and then try to separate the noise using the lifting scheme with these different weight methods. And we again saw that our proposed area prediction method is giving the smallest mean squared error in the function estimation. And in the interior region, these may seem a bit different, but the scale is very, very small. So actually they are very close to each other. So for another test function, we observe the same patterns, and our proposed methods were giving the smallest mean squared error. This was a talk about the Voronoi tessellation and how the Voronoi cells are affected by the different types of boundaries. And we then develop a method that can predict the Voronoi tessellation cell area. And we use these in the lifting scheme context to estimate the denoised values of the noisy functions. And then we found out that our proposed methods are actually giving more accurate results than the standard weight methods. So thank you for listening my talk. And if you have any questions, just drop me an email and I will be happy to answer all of them, and enjoy the rest of the conference, and see you again soon. Thank you.